Dear students, myself Dr. Viral Shukla, Assistant Professor and Head Department of Microbiology, LG Institute of Applied Sciences. I hope you all are in good mood to learn about translation. Translation process I have divided in a two-part. In the first part, we'll learn about the features of genetic code, role and structure of tRNA and rRNA. So let's start with the detailing of the translation process. As per the Gujarat University, it is a part of MI 301, Unit 2. Now, let's first understand about the translation. Now, protein coding gene expression is actually known as translation. Now, protein synthesis is known as translation because it is a decoding process. Decoding means the genetic codes present onto the mRNA that will be decoded during the process. This process is known as decoded in a form of amino acid. So that's why it is known as translation. Now, during translation, the nucleotide sequence is read in a discrete manner, like it is going to read in 3 sets. Now, 3 sets of nucleotides. Each set is going to be work as a single genetic code or a codon. Each genetic code is codes for single amino acid. So, to understand about the translation, first we have to learn about genetic code. So, in this table, all genetic codes are summarized. Now, from this, we can inspect different features of genetic code. The first feature is that you can see that each and every genetic code is around 3 letters long. Now, these letters are nucleotide bases. Each and every genetic codes are 3 letters long. The second important feature of genetic code is each and every code are going to be recognized by anticodons present onto the tRNA. The third important feature is the genetic code punctuation. Now, what do you mean by punctuation? It's like this particular genetic code, that is AUG, which is going to code for methionine. It is almost always the first codon for the initiation of translation. So that is called a punctuation. The next feature is this 3 codons, UAA, UAG, UGA. They are known as a stop codon. They are responsible for the termination of translation process. Now, these 3 codons are also known as a nonsense codons. So the next feature of the genetic code is, you can see from the table around 64 genetic codes are there. So out of 64, 61 codons are sense codon. The next feature of genetic code is degeneracy. Now, what do we mean by degeneracy? It's like you can see over here, these 4 codons are coding for serine, and these 2 are also coding for serine. Same way, these 4 codons are coding for arginine. These 2 are also coding for arginine. Means 6 codons, around 6 codons are coding for single amino acid. So this is called a degeneracy of a codon. So as I explained you about the genetic codons, same has written over here that these groups of 3 are called codons, around 61 codons for amino acids, and each of them is read to specify a certain amino acid out of 20 commonly found in protein. And AUG is known as start codon, UAA, UAG, and UGA is always known as stop codon. Now, this nucleotide sequence in a manner of 3 sets, AUG, ACG, GAG, CUU, going to code for single amino acid like methionine, threonine, glutamate, leucine, arginine. Now, to understand about the translation, we have to move forward to check about the structure and role of tRNA and rRNA. So from this figure, I hope you can understand that now what we will going to learn about. First, we'll go with the structure of tRNA. Now, from here you can see this is a structure of tRNA and this is a ribosomes. Now see, whenever translation process is going to occur, ready tRNA with a specific amino acid should be available. Then only translation process is going to be occur. Now to understand this, first we need to go with the detailing of structure of tRNA. Now this tRNA is around 70 to 95 nucleotide long, and tRNA is having different feature. The first feature of tRNA is it is having a accepting stem. Now on this accepting stem, it is always having a amino acid. This is accepting stem. It is having a active amino acid over here. The second feature of this tRNA is on 3' end of tRNA, it is always having a sequence of CCA. And on this A sequence, active amino acid is going to be attached always. Now, the third feature of tRNA is it is possess anticodons complementary to mRNA on an anticodon arm. So this is the structure of tRNA and basic features of a tRNA. Now let's understand about the ribosomes and rRNA. Now you can see from the figure, two different subunits are involved in a translation process, large subunit and small subunits. Now these subunits are having two functional domain. First domain, that is known as a translational domain, and the second domain, which is known as an exit domain. Now translational domain is always there in both the subunit, and means it is formed by both the subunit, and the exit domain solely formed by the large subunit. Now, one more thing, this subunits are actually a bench for the RNA to work on. Now, this translational subunit, or sorry, the functional site of translational site is always possess three different sites. The first site, which is known as A site, that is known as amino acyl site. Second site, which is known as peptidyl site, and the third site, which is known as exit site. Now this A site, it is known as amino acyl site or accepting site, which is going to accept an active tRNA molecule attached with an amino acid. Now this peptidyl site is going to hold a tRNA molecule with a growing polypeptide chain. This exit site, it is a location from where empty tRNA is going to leave the ribosome. Now you can see from the figure, it's like a hamburger structure. Now this particular ribosomal subunit or this particular hamburger structure is known as a workbench with a RNA blueprint. So you can understand about this particular hamburger molecule, and you can see from this slide too that ribosomes are the structure where polypeptides are built. They are made up of protein and RNA. Each ribosome has two subunits, large one and small one, and they are going to form a hamburger bun. This ribosome provides a set of handy slots where tRNA can find the matching codons, and these slots are called A, B, E sites. Now, during translation, so this particular codons are present onto the mRNA and they are going to convert it into polypeptide. Now, before concluding the session, I just want to explain you about the role of ribosomes. Now, 16S subunit of 30S ribosome is having a function that it is responsible to bind with a ladder sequence from a 3' end of its own. This ladder sequence means that is a Shine-Dalgarno sequence. Now, what is ladder sequence? During transcription, the first site or the first nucleotide sequence which is going to be transcribed, which is known as a ladder. It is also known as a Shine-Delgarno sequences. Now, this ladder sequences may be transcribed but nor be translated. Now, this ladder or Shine-Delgarno sequences are going to attach with the 3' end of 16S subunit of 30S ribosome. And so by that way, this Shine-Delgarno sequences are going to be work like a ribosomal binding site. Now, the second role of ribosome is 23rd subunit. Now, this 23rd subunit is going to work as ribozyme during the translation process. Now, what is the role of ribozyme? Ribozyme role is to add a peptide bond during the polypeptide sequence growth. So by that way, tRNA and ribosomal RNA are involved in translation process. So now let's move on the overview of translation. Translation is divided in 3 different processes: initiation, elongation, and termination. So during initiation, in this stage, the ribosomes gets together with the mRNA and first tRNA, so translation can begin. Means large subunit and small subunit get coming together, and they are going to bind with the mRNA at the Shine-Delgarno sequence. And first they will start with the translation from the start codon. Now the next is elongation. In this stage, amino acids are bound to ribosome by tRNA linked together from a chain. So tRNA will come on A site, then P site. That is elongation process. Now in termination, in the last stage, the finished polypeptide is released to go and do its job in the cell. So this particular portion we'll learn in translation part 2 in detail. Thank you, dear all. Have a nice day.